Okay, should be a bit better now. Okay, so there's 7 of you here. That's great. Hopefully a few more of you will register. We're going to go through meiosis and mitosis. Hopefully some of you will have been able to go onto the quiz room, which is at socrative.com. It's running now. It's called A-Level Biology Tutor. If you go to that now, you'll be able to log in through the student login. There's a starter quiz, which is excellent, should load automatically. And I've also put a link to the quiz room, and that's in the chat box. So you can log in directly through that. Next week, we're doing heart, blood vessels, and the cardiac cycle. If you found this useful, then please recommend it to your friends and follow me on Twitter or subscribe on YouTube. It's Understand Biology. Okay, let's get on with this. So we're going to look at mitosis, and then we're going to look at meiosis. So the first point about mitosis is it's the process by which you divide a cell into 2 genetically identical daughter cells. That is, cells that are the same as you and the same as the cells that made you. So why do cells need to divide? So firstly, they need to divide to give you more cells for asexual reproduction, and then additionally for growth. Now, students will very often write growth as an answer for why things need to do mitosis. And the answer is it's growing the organism through the production of more cells, not just growth. So growth of the organism through the production of more cells. Additionally, you need to repair damaged tissue by replacing it with non-damaged cells. Replace damaged or worn out cells. Okay. So then we have the cell cycle. Cell cycle divides into different phases. So we start off with interphase. This is a phase which is often ignored by students. It's where the cell prepares to then divide by mitosis. And mitosis itself is the splitting up of the 2 the chromatid, pulling apart of the chromatid so that each nuclei gets exactly the same DNA, because it gets one of each of the 1/2 of the chromosomes. So interphase divides into G1, which is growth, then S phase, which is the synthesis of DNA, and then in G2, which is the production of proteins necessary for the chromatids to be pulled apart. So here's the cell cycle. We've got G1, the growth of the cell, more cytoplasm, more organelles, the cell going about its normal life. Then you have S phase, which is the replication of the DNA through semi-conservative replication, where you pull apart the 2 strands of DNA and then you bring in new nucleotides. And then G2, and then into prophase, metaphase, anaphase, telophase, cytokinesis to produce 2 new cells. Okay. Again, G1, protein synthesis, increasing the amount of cytoplasm, replication of organelles, synthesis of the DNA, followed by G2, making the proteins necessary for cell division. Now, DNA, remember, is in the nucleus. Long thread-like DNA in a non-dividing cell is called chromatin, and this replicates to form 2 chromosomes, to form a chromosome made of 2 chromatids. So just to show you this on a whiteboard to make my point. So you came from a sperm which met an egg, and the sperm had an N number of chromosomes. It was haploid, and that met an egg which was also haploid, and that produced a diploid cell. Diploid cell is 2N, and this is the most improbable event of your existence. It's the event that your parents are most famous for, producing you, of fertilization. Now that cell starts off an N, come together, fertilize with another N cell, so 2 gametes forming a diploid zygote. If we just look at 1 chromosome in each, so we've got 1 red and 1 yellow chromosome. Those are going to come together and you're going to end up with 2 chromosomes. Now, what then happens is interphase, and this is the bit that confuses quite a few students, because interphase the DNA looks like this. It's not condensed, and it's being used for protein synthesis. And then S phase, it replicates. And remember, in S phase, you've actually got two complementary strands of DNA, and those two strands are pulled apart, and then new nucleotides come in from digestion, in your case, and in plants they're made by joining them together with nitrates and phosphates and carbon dioxide from the air. And then those coil up and 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 condense. And then you have a centromere holding the two chromatids together. And the two chromatids are genetically identical copies of each other, because one chromatid contains one old strand and one new strand that's come from DNA. Well, done from DNA from digestion. So if we were to put a gene on these, so we had a gene here, so we have the A gene, and then we had, say, the little a gene on the homologous chromosome. These two genes are at gene loci. Now, when they replicate, you're going to get two genetically identical chromatids forming a chromosome. So when you see them in prophase, you see the replicated chromosome as a chromosome, and that chromosome is made of two chromatids. So this cell is still 2N. This is a huge point of misunderstanding by students. This is a 2N cell with two chromosomes, and during interphase it replicates to give you another 2N cell with two chromosomes in it. So we've doubled the quantity of DNA, but the quantity of information has remained the same. If you had two copies of a recipe book, you've still got the same number of recipes. So the process in interphase is the replicating of the DNA to give you two identical chromatids. Now, this is how they appear in prophase, and then we have the next phase, which is metaphase. So if we do a few circles for this, so in metaphase they're going to line up on the middle. Metaphase, think the middle. In anaphase, they're going to get pulled apart. And in telophase, you're then going to form two new nuclear membranes. Now remember, we started off with two chromosomes. Each chromatid is going to be pulled apart. When each chromatid is pulled apart, it's then referred to as a chromosome. Now they're pulled apart by being pulled by spindle fibers. These are proteins that grow out from the centrioles and pull apart the chromatids at the centromere, and then pulling them apart to opposite poles of the cell. And then we form a new nuclear membrane around the outside, and then the cell splits in a process of cytokinesis, where they split down the center of the cell. And then we end up with two cells, both of which are going to contain two chromosomes. We're going to contain a red one in each and a yellow one in each. Now remember, we started off by putting the A gene on it. Remember, we're going to have exactly the same as we did in the parent cell. So through this process of mitosis, we'll have produced two genetically identical cells, which are identical to each other and identical to the parents. Okay, let's go back to the PowerPoint. Any questions at all, just put them into the Q&A box and that will pop up, and hopefully we can answer them as they go along. So we've got two parts. We've got a chromatid and a centromere. The chromatid is two chromatids are held together by a centromere. So this is the step that occurs during S phase of interphase, is that we have duplication, and then they coil up to give you the two chromosomes, two chromatids, which are genetically identical. Again, making this point, we go from this to duplicated like this. So genetically identical sister chromatids. Exact copy of each other, held together at the centromere. Here are two homologous chromosomes. Remember, homologous chromosomes contain the same genes, and they may have different alleles of those genes, but they contain the same genes at the same positions. The replicated chromatids are going to be genetically identical to each other. I think I've made this point enough times. I hope it's starting to go through to you. Okay, so all organisms have their own specific number of chromosomes. We have 23 pairs. Dogs have 78 chromosomes or 39, goldfish, lettuces, various numbers. So these are all the number that you have in a somatic cell, and a somatic cell is all of your cells apart from your gametes. So all cells apart from sperm, eggs, or pollen. So every single cell in you has got 46 chromosomes apart from your sperm and your eggs, and the same with the fruit fly. Every single cell has got 8 chromosomes apart from fruit fly, sperm, and eggs. So we have interphase, where the cell grows and then prepares to divide. DNA gets replicated. And then we have prophase, metaphase, anaphase, telophase. So remember, the way to think about this is in Portsmouth, men are tall. Interphase, prophase, metaphase, anaphase, telophase. Prophase, the chromosomes condense and appear. Metaphase, they line up on the middle, on the equator. Anaphase, they get pulled apart. And in telophase, they then form two, with 2 new nuclear membranes around the outside. So prophase, metaphase, anaphase, and telophase. Here's prophase. The chromosomes condense and coil up. Nuclear envelope disappears. Nucleolus disappears. Remember, the nucleolus is where the ribosomes are made. Centrioles move to opposite poles. Spindle fibers form. Spindle fibers join onto the centromere. Prophase, you see this is a cell in late prophase, and this is later prophase. Here we just start to see these irregular sort of granular shapes in the nucleus. Metaphase, they line up on the middle, arrange themselves on the equator, form microtubules. Anaphase get pulled apart. That's metaphase. Anaphase, the centromere splits, the 2 chromatids get pulled apart, so each chromatid therefore becomes a chromosome. Pull them to opposite poles. Each chromatid is now called a chromosome. Here's anaphase again. Telophase, forming 2 new nuclear membranes around a complete set of 2N diploid chromosomes. Nuclear envelope reforms, 2 new nuclei formed, spindle fibers disintegrate. So here we've got the 2 new nuclear membranes forming, and then we're just starting to see a cell plate form down the middle. Then we have cytokinesis. Now in you, it's called cell cleavage, and in plants, it forms a end plate, well, cell plate, sorry, on end plate. And then it carries on back as normal, goes back to interphase and carries on growing. So this is a cell dividing, this is an animal cell dividing by cleavage. Remember, this is the first thing that you did when you went from being a 2N fertilized zygote to forming 2N cells. This was you at your earliest form. And it's happening in a plant cell where we form cell wall down the center, and then we form 2 daughter cells separated by a cell wall. So remember, interphase, don't forget it, we've got G1, then S phase, which is the replication of the DNA, so the quantity of DNA doubles, and then we have G2, and then we have mitosis, which is prophase, metaphase, anaphase, telophase, then we follow it with cytokinesis. If you're asked to identify things, if you're asked to identify different stages, and often this comes up as an exam question, most of the cells you're going to see most of the time are in interphase. Cells spend 80 to 90% of their time in the interphase. So you should expect to see, if you look at a cross-section through, say, a root tip cell, you should expect to see most of the cells in interphase. When you start to see the chromosomes appear, that's in prophase. When you can see them distinctively lined up on the middle, that's metaphase. When you see them being pulled apart, that's anaphase. And when you start to see two new nuclear membranes forming, that's telophase. I'll go through this. I think I've made all the points. Why is it important, mitosis? It gives you the production of genetically identical cells to each other and genetically identical cells to the parents. Enables things to grow by producing more cells. Enables you to repair through replacement of damaged or dead cells. Asexual reproduction, this gives you complete offspring which are identical to the parents, so this takes place in yeast and bacteria, and also in flowering plants, some flowering plants where they produce rhizomes or bulbs or tubers that enable them to reproduce asexually. Okay, now's the time to ask any questions in the Q&A slot. I've got one question there, and then I've got the next quiz to put up, which I will put up now. So if I go to Socrative quiz and I launch another quiz. Okay, so the next quiz is running. So if you'd like to have a go at that, it's at Socrative, alevelbiologytutor.com. Sorry, Socrative, if you put A-level biology tutor in. A couple of people giving answers already. Can you explain the difference between sister chromatids and normal? I'll give it a go in a minute. I'll just paste in the link. So the link to the quiz is in the chat room as well. What happens during prometaphase, says Alex, and how do you differentiate between an image of telophase and cytokinesis? Well, what happens during prometaphase is where the chromosomes are moving from being in prophase into metaphase. Part of the problem is that you're dividing up a cycle that's occurring continuously, and you're dividing it into artificially. So in prometaphase, the chromosomes are condensing and then moving towards, or being moved towards, the equator. I hope that answers your question, Alex. And Selena says, how do you differentiate between an image of telophase and cytokinesis? Well, if it's a plant cell, you'll see cell cleavage. You'll see the two cells, the cytoplasm will be pinching off to give cytoplasm in one cell and cytoplasm in the other cell. And then in cytokinesis of a plant cell, you'll see the formation of a plate a cell plate between the two cells. I hope that answered your questions. And Emma, you said, can you explain the difference between sister chromatids and normal? A sister chromatid is when there are two chromatids, each forming, well, they form a chromosome. Each one has been produced by the semi-conservative replication of a chromosome. So two sister chromatids are genetically identical because they each contain one old strand of DNA and one new strand of DNA. So I hope that explains to you, Emma. Okay, let's see how you're getting on with the quiz. Quite a few going. Well done. Lots of good people doing well in the quiz? There's a link to the quiz in the chat room, and there's also, if you go to socrative.com into the student login and put in A-level biology tutor. Okay, I'll give you about another minute or so to do the rest of the quiz. Any further questions, people, before I move on? Please feel free to ask them. Okay, looks like most people have finished the quiz, so I will move on. And what we're going to move on to do now is to talk about the differences between mitosis and meiosis. So, first difference is that mitosis produces two end cells, those two diploid cells, and meiosis has 2 rounds of division. So mitosis has 1 round of division and mitosis has 2. Remember, in mitosis, you're going from a 2 end cell to 2 end cells and then to 4 end cells through another round of division. Mitosis, you have the same number of chromosomes. Meiosis, you halve them. Mitosis, all cells are genetically identical to each other and to the parents, whereas in meiosis, the daughter cells are genetically different from each other and from the parents. In mitosis, the homologous chromosomes don't form pairs, and in meiosis they do. They form pairs on the equator in metaphase. There's no crossing over between homologous chromosomes because homologous chromosomes don't form in mitosis, whereas crossing over does occur where genetic information is exchanged between maternal and paternal chromosomes as a result of the formation of chiasmata. And chiasmata is where the chromosomes break off. So on to chromosome number. So we've all got 46 chromosomes, and we contribute 23 from each parent. So you got 22 from your dad and an X or a Y chromosome for a boy, and you got 22 from your mum and an X chromosome. This is a diploid cell, so we've got 2 chromosome 2s all the way down to the end of the autosomes, which is chromosome 22. So this is a karyotype that's taken from a boy, and we know that because we've got an X and a Y chromosome. So if it was a girl, we'd have 2 X chromosomes, but this is from a boy because we've got an X and a Y. Now, meiosis, the principle of meiosis is that you put 1 of each of the 2 chromosomes into the gametes, and we're going to look at how that occurs. So we start off with the concept of a homologous chromosome. Homologous chromosomes is what you got 1 of from each parent. So they're the same length, centromere is in the same position, and they carry the same genes that control the same traits. And those genes are carried at a loci or a locus, which is a position of a gene on a chromosome. Now, you may be homozygous for a particular gene, which means you contain 2 of the same alleles, or you may be heterozygous for a particular gene. So hetero means different, so heterozygous, different in the zygote, and the zygote is your first diploid cell that formed you. Talk through what chromosomes and chromatids. So this is a chromosome that's made of a chromosome that has been replicated to form 2 chromatids forming a chromosome. This is a chromosome, and this is a pair of homologous chromosomes. Process of meiosis, we go from a 2-end cell, we then go through interphase, prophase, metaphase, anaphase, telophase 1. So we still have interphase, which involves G1, S phase, and G2. So remember, we're still replicating DNA semi-conservatively in S phase. Then we go through prophase, metaphase, anaphase, telophase, and we produce two end cells. Then, and we do that by taking one of each pair of chromosomes, we pull them apart. We pull apart the homologous pairs so that the cell at the end of meiosis I has got one of each of the pairs of homologous chromosomes. So you're putting, so if this was the if this was the chromosomes, if that's the chromosome for your mum, then you either put the chromosome from your mum or you put the chromosome from your dad into the cell at the end of meiosis I. Now, meiosis II is a mitotic division where you just pull apart the chromosome exactly the same as in mitosis. And when you pull the chromosome apart, each chromatid becomes a chromosome and ends up in the daughter cells at the end of it, forming 4 haploid gametes. So with mitosis, you've got 1 round of replication and 1 round of division. Whereas in meiosis, you've got 1 round of replication and 2 rounds of division. So emphasizing what's going on again, to look at the big picture, this is your 2N cell. Then you undergo S phase and then into prophase, and then they line up in homologous pairs. So here is a homologous pair of chromosomes. We can have crossing over at this stage of prophase. Then one of each of the homologous pairs is pulled to opposite directions, then the cell splits, and then we pull apart the chromosome, the chromatid split at the centromere, and they're pulled to opposite poles, and we end up with 4 end cells. So the purpose of it is that we produce cells containing half the original number of chromosomes, 2 stages, meiosis I and meiosis II. And how does it produce variation in the gametes? It produces variation in the gametes through crossing over of the homologous pairs, random assortment of the homologous chromosomes, and then in terms of sexual reproduction, it gives you haploid gametes for random fertilization, random fusion of gametes. So interphase, standard interphase, replication of the DNA, formation of new organelles in G1, protein synthesis, ATP synthesis, replicate the mitochondria. Then prophase. So prophase 1, we get the condensing of the chromatin, forming visible chromosomes. And now each homologous pair of chromosomes come together in a process called synapsis, and this forms a bivalent or bivalent. Now, later on in prophase is where we get the formation of chiasma, and chiasma are where we get the non-sister chromatids exchanging genetic information. So this chromatid and this chromatid will contain the same genes, but they will have exchanged different alleles. Now, I will talk about that more later. So the process is, the point at which it crosses over is called a chiasma. The plural is chiasmata. And this exchange gives you a chromatid, which is a hybrid of maternal and paternal DNA, because you've got exchange between the homologous chromosomes. So here's another example. So just to show you with a laser pointer, so we've got a sister chromatid containing the A and the A, big A and the big A. Remember, these 2 chromatids have been produced by semi-conservative replication, so they're genetically identical. When they line up in prophase, they then form chiasma and can cross over. So now we're going to have a new combination of alleles, because we're now going to have this chromatid will contain some of the other chromosomes alleles, because it will have been swapped over or crossed over. So synapsis is where they pair up, they then cross over, Then we have metaphase 1. Metaphase 1, the bivalents line up on the equator. And what's important to remember here is they don't line up in any particular order. So you don't have the maternal ones on one side and the paternal ones on the other side. You have them lining up in pairs of homologous chromosomes, but there is no order. It's not like a folk dance where all of the girls on one side and all of the boys on the other. It's an independent assortment. It's a random process. So the chromosomes arrange themselves randomly which side they're on. So they're always in homologous pairs, but which side they're on is random. So this is trying to make the point. So if you look here, we've got a homologous pair of chromosomes, and here's another homologous pair of chromosomes. And then over here is the other option. So if I just annotate this, so here we've got the little b and the S genes. So this is the little b here, and here is big B there and there. Now, the two chromatids are genetically identical because they've been produced by semi-conservative replication. When we pull these ones this way and these ones this way, we're going to produce that in one gamete and that in the other gamete. But because this one over here, they've lined up in a different order, we're then going to produce that in one gamete, and we're going to produce that in the other gamete. Now, you've got 23 pairs of chromosomes, which means you've got 2 options for each chromosome 23 times. So the number of different options you have is 2 times 2 times 2, 23 times, which is some 8 million different options through independent assortment. So independent assortment halves the number of chromosomes when the homologous pairs are pulled apart, but it also gives you genetic variation because you either put your dad's chromosome 1 into the daughter cell, or you put your mum's chromosome 1 into the daughter cell. And that happens obviously with 23 pairs, so it gives you a tremendous amount of variation. Here's more points to make about independent assortment. So this is if you lined up all of the maternal on one side and all of the paternal on the other. This is chromosome 1, 2, and 3. This would give you different options from this one, and from this one. So each one of these is going to give you a different combination of chromosomes in the daughter cell, so they're each going to give you more genetic variation. Okay, so metaphase 1, the homologous pairs line up on the equator. Then they get pulled apart. So in anaphase, you're pulling the centromere towards the pole. And remember, you still have here a chromosome that's made of 2 chromatids joined together by a centromere. So you're pulling apart the homologous chromosomes. They go to the opposite poles, and then you exactly the same as mitosis, you go through telophase and cytokinesis. But in this case, you're forming 2 new haploid cells, because you've gone from having in you 46 chromosomes, and the chromosomes look like this, so you've got pairs of homologous chromosomes, to putting 1 of each pair in each daughter cell. So they're now haploid. They've got 23 chromosomes in each. But remember, the chromosomes are still made of 2 chromatids. Now, these daughter cells are now genetically different from each other, and they're also genetically different from the parents. So going through meiosis I again, remember we've had interphase, so we've had the replication of the chromosomes. Metaphase, they line up on the middle in homologous pairs, and independent assortment occurs. Anaphase 1, 1 of each of the homologous pairs gets pulled each way, and then we form telophase and then cytokinesis, and we've gone from a 2N cell where there were 4 chromosomes to an N cell where there are 2 chromosomes. These chromosomes then go into meiosis II, where they get pulled apart, in another division, which is a mitotic division, where you're pulling apart the chromosome, pulling apart the two chromatids of the chromosome, so they form separate chromosomes. Good. Right, let's plow on. Next thing, I've got true, false questions on meiosis. So if I just go and start that quiz for you. So I've got all of you getting into the room, that's good. And there's 10 or 8 true, false questions. Some of you are starting to answer them already, which is terrific. If anybody has any more questions, then please do feel free to ask them in the question and answer chat box. I'm just putting the link to the quiz again into the webinar chat if anybody's joined late. Anybody got any more questions? Feel free to ask them. I've got a question up in the chat box. So, what does bivalent mean? Well, Emma, bivalent means a pair of homologous chromosomes. So if I just go to whiteboard and try and make the point to you. So you've got, remember we started with a sperm with 1 chromosome, and we started with an egg with, again, 1 chromosome. These come together and we produce a cell which has got 2 chromosomes in it. It's 2N. Now that grows and grows and grows and grows and grows by mitosis. Then at some point you're going to start meiosis, and in meiosis, in prophase, remember that the chromosomes have replicated, and in prophase they coil up and they appear, and the bivalent is the pair of homologous chromosomes. So this would be, for instance, a chromosome 4 or something from each parent, and this chromosome 4 lines up with the other chromosome 4 from the other parent, and they then go to the equator of the cell, they line up in metaphase, and then they get pulled apart. Hope that's answered your question, Emma. Okay, lots of you seem to have done the quiz, which is excellent. A couple of you just taking your time over it, and I'll go. A question up here. Do we need to know the difference between plant and animal cell division? Well, the only difference between plant and animal cell division is that firstly, you have the difference with the centrioles. So in animals, you have centrioles, and in plants, you have centrosomes. And centrosomes are where the spindle fibers grow out from, but you can't see them, whereas in animals, you can see them as centrioles. The division is exactly the same process, except with you've got cleavage in animal cells, and you've got the formation of a cell plate in plant cells. but the process is exactly the same. I hope that answered your question, Ravi. Okay, I'll quickly plow on to the next bit. So the next bit, remember, is my Meiosis II, and this is a mitotic division. So what happens now is that the centrioles replicate. It forms another spindle at right angles to the original spindle. The nuclear membrane disappears, the nucleolus disappears, and then the chromosomes get pulled apart, exactly like mitosis. So here we've got one of each pair of homologous chromosomes, because remember, you've halved the chromosome number, so the cells now in Meiosis II are haploid. So you've only got one of each of the pair of homologous chromosomes. Now they line up randomly on the equator. Due to the way they line up on the equator, because remember, the sister chromatids are not going to be identical now, because they're genetically different from each other because of crossing over that's occurred in prophase 1. So when you've got the sister chromatids in mitosis, they're genetically identical because there's no crossing over in mitosis, whereas in meiosis, in Meiosis II, the sister chromatids are going to be genetically different from each other because crossing over will have occurred. Now I see, do I have somebody have a hand up there? Nope, can't see. Okay, keep going on. Anybody's got any questions, just put them into the Q&A box and I'll address them. So the way in which the chromatids are aligned on the equator will give you genetic variation when those chromatids then get pulled apart in anaphase 2. So the centromeres divide, the spindle fibers pull apart the chromatids, which are now intact chromosomes, and we've then gone from an N cell to 2 more N cells. Because we took a chromosome, we've pulled it apart, and we've now put a chromosome in each cell. So in Meiosis II, the number of chromosomes remains the same, but the quantity of DNA in each cell halves. So here's anaphase 2. So we've got here forming 4 N cells, and then the telophase will occur, forming 4 new nuclei. So cytokinesis begins, forms 4 haploid cells. The chromosomes are genetically different from each other, and then the DNA will start to unravel, and you finish the end of the process. So here's telophase 2. Running through meiosis again, remember, having finished meiosis I, you then end up with 2 haploid cells, each of which contains one of the 2 homologous pairs. These then line up on the equator. The orientation of the chromatids matters because the sister chromatids are now not genetically identical. They then get pulled apart again. Between here, we've got 2 chromosomes, and we end up with 2 chromosomes in each cell. So we've gone from 2 N cells to now 4 N cells. Between here and here, we've halved the quantity of DNA, but the number of chromosomes has remained the same. A few points about crossing over. Now, when they cross over, you can produce, obviously go from 1 to 2 to 3 to produce these crossed over. Now, this is a pair of homologous chromosomes. We've had exchange of information. Now, when these line up on the equator of the cell in metaphase 2, the way they're orientated matters, because if they're that way round, then one half will go one way, one half will go the other way. If they're the other way round, then the other half will go the one way and the other half will go the other way. And that will give you also genetic variation in the gametes. Okay, so processes by which you get variation. You have crossing over in prophase 1, where you get the non-sister chromatids form chiasma, break and reattach to each other, and that process is often called recombination. Now that gives you a new combination of genes. Well, no, it doesn't. It gives you a new combination of alleles on each given chromosome. So each chromosome is therefore a mixture of maternal and paternal, not genes, alleles, or genes from each maternal and paternal chromosome. Next one, independent assortment of homologous chromosomes. As a result of them lining up randomly in metaphase 1, they get pulled apart. So you either get your mum's chromosome 1 in your sperm, or your dad's chromosome 1, or 2, or 3, or 4, or 5, but you don't get both of them. And that means that each of the daughter cells, and daughter cells can be the sperm, the eggs, or the pollen, will contain a different combination of the maternal and the paternal chromosomes. Independent assortment of chromatids during metaphase 2. The sister chromatids are not genetically identical because they've crossed over. So the way that they align determines how they segregate and separate in anaphase 2. And it produces genetically different gametes. Okay. Just try and explain that point about the non-identical chromatids. I'll go up to the quiz, not the quiz, I'll go up to the whiteboard and we'll try and explain that, because I think that's a point where I might have lost a couple of you. So let's have a look at this again. So let's make this a bit bigger. So we have one chromosome which is replicated in prophase, and then we've got another chromosome that is replicated in prophase. Now these then line up in so they're replicated in interphase, they line up forming ambivalent in prophase, and then crossing over occurs. Now, crossing over will give you a hybrid chromosome, and then they're going to get pulled to opposite poles, and then we're going to produce 2 daughter cells. Now this one is going to have a bit of that bit there. Now, what matters now in metaphase 2 is which way up these are. Because if it's this way up, then this one and this one will go this way, and this one and this one will go this way on the when they get pulled apart. But if it's the other way up, if it's turned round, then this chromatid will get pulled this way, and this chromatid will get pulled this way. And remember, in you're not dealing with one chromosome, you'll be dealing with 23 chromosomes. So the orientation of the chromosomes in metaphase 2 is really important in terms of the non-genetically identical chromatids getting pulled one way or the other. Unfortunately, I didn't do a very good PowerPoint on that, or I should have done. So Axel says, I'm confused how a parent cell forms n daughter cells in meiosis II. Why is it not half n if it has divided? I presume what you mean is it's divided again. Well, the point is that the cell divides, but the chromosome number doesn't change, Axel. So let's just show you this again on a picture. So please feel free to keep answering questions. So we start with a chromosome from one parent and a chromosome from the other parent. These replicate in interphase, form the homologous pair, then they get pulled apart. So we start with a with 2 chromosomes, and then when they've been pulled apart, you're going to have 1 chromosome in each, and then they're going to get pulled apart again in Meiosis II, and you've still got just 1 chromosome. So in Meiosis I, you halve the number of chromosomes in each cell, but in Meiosis II, the number of chromosomes remains the same. So we have 1 chromosome at the end of Meiosis I, and then we've got 1 chromosome at the end of Meiosis II. So the quantity of DNA has halved between the end of Meiosis I and the end of Meiosis II, but the quantity of chromosomes has remained the same. So at the end of Meiosis I, you've got N cells, and then at the end of Meiosis II, they're still N cells because they've still got the same number of chromosomes. Did that answer your question, Axel? Hope it did. Okay, on to the next one. So, comparing the two. Remember, mitosis results in the production of 2 genetically identical diploid cells, whereas meiosis produces genetically different haploid cells. Mitosis produces 2, meiosis produces 4. Mitosis, all of the chromosomes and all of the alleles are identical, whereas in meiosis, the alleles and the chromosomes are genetically different from the parent diploid cell and also from each other. Any questions, feel free to ask them. I've got a final and much longer quiz for you all to enjoy, which is a bit more difficult. So if I go and start that running, and then I can answer some questions. Okay, I've started the final quiz running, so I hope you can enjoy that. And I will now answer any questions that you've got. Axel says, yes, I understand it now. Thank you. Excellent, Axel. That's awesome. I'm delighted that you understand it now. That has made my evening. Okay, I'll leave this all running. Any more questions, people? Feel free to ask them. I'm here to help. Okay, so the final quiz is running now. If you go to Socrative, if you haven't logged in already, it's A-Level Biology Tutor is the student login, and also I put the put it into the chat box. I put the link to the quiz. I'll put it in again if you've missed it. Next week's webinar is on heart, the cardiac cycle, blood vessels, blood flow. Please register for next week's at www.alevelbiologytutor.com. If you found it useful, then do tell your friends and invite them, because everybody's welcome. And if you want to subscribe, then I'm on Twitter at Understand Biology, or on YouTube at, again, Understand Biology. I've got a YouTube channel with a few subscribers. Any more questions, people? Last chance for any questions. Okay, well, I hope you're happy with how things have gone. If anybody has any helpful comments, then please feel free to email me. Look forward to seeing you at the next webinar. I'll leave the quiz running so you can do that in your own time. And thank you very much. And I hope to see some of you or all of you next week. Cheers. Bye-bye.